Hi, everybody. It's Mr. Vallejo. Welcome to biology class. Today we'll be taking a look at Mendel's laws of inheritance. Let's take a look at genetics. So let's go ahead and share that screen with you. I'd like to remind you that these documents, this PowerPoint, is available for you in your learning management system, whether that is Schoology or Canvas. Let's go ahead and begin. All righty. So before we even start with our study of Mendel's laws, we've got a few things to define. So before we even do that, why should we be interested in inheritance or heredity? Well, let's take a look at this one. It says purebreds and mutts, the difference of heredity. Purebred dogs are very similar on a genetic level due to selective breeding. And aren't those cute puppies? But I've always gotten my dogs from the pound, and they were rescue dogs. They look a lot like this dog, kind of mixed all over the place. And so in my adult life, I've had 6 Labrador Retriever mixes, and they're all mutts. They all look a little bit different from each other. Mutts or mixed breed dogs, on the other hand, show considerably more genetic variation. So that is interesting. Mendel's laws. The historical roots of genetics, the science of heredity, has ancient roots. Get it? Back to ancient attempts at selective breeding. This is wild. I had broccoli last night. I kind of like broccoli. Um, Cabbage has a lot of flavor to it. Kale. Well, more and more people are eating kale these days. Remember 20, 30 years ago, you wouldn't touch that stuff. Speaking of 30 years ago, when I was riding around in a car with my mom in the backwoods of Salinas, California, every once in a while we'd be driving behind a produce truck and it would drop some of its load, and my mom would pull over and she would say, "Son, go and get that cauliflower for dinner." And I would bust out of the car and go grab the cauliflower. So sometimes I like cauliflower, sometimes not so much. But all of these plants have their ancient roots in the wild mustard plant. That's pretty interesting. So how do we get that? Well, what you do is you take the characteristic that you like and put together a male and a female, and keep doing that for a long hundreds, a long thousands, a long, long time. And you'll see that after a while, you get some of the characteristics that you're interested in. Before we get into the genetics, here's some stuff we need to know. We're going to go over some of these genetics terms. These are all related to each other, so that's why they're paired up the way that they are. And after we do this, then we can have a better understanding of Mendel's laws. Speaking of Mendel, Mendel is known as the father of genetics. And it says here, the experimental genetics began in an abbey garden with quantitative experiments with pea plants. Now, people knew through practical experience that when you got two really hairy dogs and you put them together, and you got some hairy puppies. Or if you took some plants and kept uh, taking out the ones that have a particular characteristic, after a while you get some broccoli. People knew that. But the difference with Mendel's experiments is that he was able to produce quantitative data. He was able to give us some numbers. He had over 4,000 plants that he took care of in his garden. And he took meticulous notes, and he noted what the organisms look like when you crossed, say, a purple flower with another purple flower, or a purple flower with a white flower, or a pea plant that had round peas, smooth peas versus wrinkled peas, and that type of information. So Mendel crossed pea plants that differed in certain characteristics and traced traits from generation to generation. Here's a purple flower, here's a white flower. And when you take the sperm from this white flower that's a pollen, and you put on the female part, it's called the pistil of this purple flower, then each of these individual peas in the pod are all different from each other, but they all produce purple flowers. What happened to the white one? We'll see in a little bit what happened to that white characteristic. But in this case, all purple. Mendel hypothesized that there are different forms of genes. We know that from our study of mitosis and meiosis already, that there are different forms of the same gene on homologous chromosomes. Genes are the units of heredity that determine inherited characteristics. Now, it's interesting that Mendel knew all of this without knowing anything about chromosomes. And he was able to derive this because he had very good quantitative information. So you take a look at the table here before we leave this slide. These are some of the characteristics that he was able to examine. And you can see the different forms or different alleles or alternate forms, as I wrote here, of the same gene. So flower color comes in purple and white. Pod shape comes in inflated and constricted. Seed color comes in yellow and green. So the thing is, Mendel's mathematics worked out swimmingly. And so sometimes it has been questioned whether perhaps Mendel had some divine help, because these 7 characteristics happen to be on 7 different chromosomes. If any 2 of these characteristics were on the same chromosome, then his mathematics wouldn't have worked out, and we would not know him historically as the father of genetics. So before I knew how to do Photoshop, I have a picture of Mendel right here being lifted up by a cloud that looks an awful like a hand lifting Mendel up. Did he have divine help? I don't know, but let's take a look at some of the terms that we need to know in order to speak intelligently about genetics. First, we have locus, allele, and pair. Now, some of these we've seen before. A locus is the physical location of the gene on a chromosome, as we studied in our mitosis and meiosis discussions. And the physical location of the gene, the locus, is where you might find different alleles. Different alleles are forms of the very same gene. You're going to have one of those alleles from your mother and one of those alleles from your father. So you get two copies of the gene, one copy from the mother, one copy from the father, and they may be the same form, the same allele, or they may be different forms or different alleles of the same gene. But there's always 2, so there must be a pair of alleles working together, a pair of copies of the genes working together to actually show the characteristic. True breeding individuals for a certain characteristic always have offspring with that trait. That's why when we saw the purple flower previously, back over here, this purple flower always has purple kids. And so we say that purple flower is true breeding or pure breeding, or another term for it, as we'll see, is homozygous. True breeding individuals for a certain characteristic always have offspring with that trait. But sometimes you have a hybrid or carrier that has 2 different copies, two different forms of the same gene, 2 different alleles. One form does not show, but the other one can still be, but that one that doesn't show can still be inherited. Now, these are similar terms. Homozygous means true breeding. True breeding individuals for a certain characteristic always have offspring with that trait. That's the same thing as homozygous. Homozygous individuals for a trait have 2 of the same allele, whether that allele is dominant or recessive. We'll look at those terms in a moment. A heterozygote, remember hetero means different, Heterozygotes have two different alleles for the gene. They are also called hybrids or carriers. What? Hybrids or carriers. So homozygous is the same as true breeding, and heterozygous is the same as a hybrid or carrier. Now, dominant recessive are terms you hear a lot in genetics. The dominant allele overpowers the recessive allele so that the dominant shows in the individual. So the shorthand way to write this is with a capital letter. And then the recessive can only show in the individual if there are two copies of the recessive allele. And those are going to be shorthand, lowercase letters. So if we take a look at our previous example, we might have a purple flower that is dominant. The purple trait is dominant, so we would use a capital P. And then the recessive characteristic is white, and it only is white if you have two copies of that recessive gene. We would shorthand, we would abbreviate, we would designate that as a little p. So when you have little p, that's a recessive white flower. A dominant would be big P. That's a homozygous dominant individual. But sometimes you also have a mix. And remember, that's a hybrid or the carrier with one big P and one little p. Now, these two-letter combinations are called the genotype. The genotype is just that, representing the two alleles for the trait in question in an individual. Now, a similar term is phenotype. The phenotype is the expressed characteristic for that particular gene. We used to say that it was the outward appearance. It's not true for everything. It might be for hair color and eye color or skin color or something like that, or whether you have a cleft chin with a little indentation right there, or whether you have free or attached earlobes. You know, that's your phenotype, but it's not always something that you can see on the outside of an individual's body. I am lactose intolerant, but you can't tell just looking at me. So the phenotype is the express characteristic, we say, for that particular gene. It might be whether a person makes a particular protein or not, and that would be the express characteristic. All right, so now we're ready to take a look at Mendel's laws. Here's the first one, the law of segregation. The law of segregation is for one trait. And from his experimental data, Mendel deduced that an organism has two alleles for each inherited characteristic and one from each parent. There's two copies of the gene. They may or may not be different alleles, but it's gonna have, each person's gonna have one from each parent. And so if you take a look at the example here, there's a purple parent that's giving a big P and a white flowered parent that's giving a little p. All of these guys are going to be big P from the purple and little p from the white. These are going to have the genotype of big P, little p, but they're still going to be purple in their phenotype. But if we were to mate a big P, little p F1 generation plant with another F1 generation plant, which is also big P, little p, then what you get is this 3-to-1 combination that you see down here. Okay, 3 purple and 1 white. All right, so dominant and recessive. Here's a slide that just says it a slightly different way. One of the alleles determines the organism's appearance and it's called dominant. The other has no noticeable effect, and that's recessive, unless there are two copies of the recessive alleles. But this slide is for the situation if the two alleles of an inherited pair differ. Mendel's laws of segregation predict that allele pairs separate. That's what we mean by segregation. They separate from each other during gamete production. So these letters represent characteristics, genes that are on the gametes or in the gametes. They're on the chromosomes. And what you can see here is the situation that I just highlighted for you. Purple, big P, white, little p. All the F1 generations are big P, little p. The next generation, called the F2, the second filial generation, are going to be 3 to 1, purple to white. Why? Because if you take all the different combinations, this is called the Punnett square. We'll see that in a little bit, but you just fill this in like you would a multiplication table. There's a big P on the sperm, there's a big P on the egg. Put them together, you get big P, big P. This one's definitely purple. These two are a combination of big P and little p. So you get big P and little p, and that is then purple as well in the phenotype, but the genotypes are different. These are heterozygotes. These are hybrids. These are carriers. And then you have a 1 out of 4 chance of getting 2 little p's together. That's a recessive characteristic. That's a homozygous recessive individual. That individuals, it shows white flowers in the phenotype. So I'll remind you, because we've seen this slide before, that these different forms of the same gene, these alleles, are on the same exact location, the same locus. And so you have a homologous pair of chromosomes coming together, homologs coming together. What you have is those different genes. Here they're the same allele, big P and a big P. Same here, but they're recessive, little a and little a. But over here, you have 2 different alleles for the same gene, one big B, one little b. That results in a hybrid or a heterozygous individual. Now, before we go any further, and before we take a look at genetics problems, we need to take a look at some laws of probability. Mendel's laws reflect the rules of probability. Here's the first one. If I had a penny and I was flipping that penny and I got heads, well, that's a 50-50 chance I'm going to get that head. So, but if I flip it again, heads. And I flip it again, and I flip it again, and I flip it again, and I flip it 9 times in a row, and I get heads. Woo! What's the chance that I'm going to get heads that 10th time? I think it's a pretty good chance. Ooh, careful now, because what happens in probability is the probability of events are independent of each other. There's only 2 sides of the penny. One's heads, one's tails. So there's always a 50-50 chance that you're going to get heads or tails. Well, the probability of these events are independent of each other. So even though I flip 9 heads in a row, I still have a 50-50 chance of getting a heads that next time. Here's some other examples. I remember a time when my sister and I went to Las Vegas, and I was playing my nickel and my penny machines. But my sister, woo, she's a big gambler. What my sister did, she went to the machines, the slot machines. There you'd have to go to the cashier and give them some paper money to buy tokens that were worth $1, $2, $5, and she'd play those machines. And sometimes she'd win, and sometimes she'd lose. But the casinos would always offer my sister free rooms because she gambled so much. So one time I was with her in Las Vegas. I was playing my pennies and my nickels, and she was doing her thing. She was playing her slot machines, her $2 machines, her $5 machines. And one time I hear this, and the lights were lighting up on one side of the casino, and it was my sister. My sister had scored a big win, and it was before the electronics, but there were coins flipping out of that machine. And so the cocktail waitress came, and she said, "May I help you with your winnings?" And my sister said, "Of course." And then she's cocktail waitress put all the coins in a tray for my sister. It's about this big. It was heavy. I know it was heavy because my sister handed it to me, and she said, "Hey, hold this for me. And whatever I do, whatever I say, don't give it back to me. Woo! So I went away. I went back to my nickels and my pennies. But 22 minutes later, my sister came back and said, "Hey, I'm on a roll. Let me have the money back because I'm gonna play and I'm gonna win big." Dude, I thought she had already won big. Well, what happened was that one time she uh, she lost it all again. But I'm telling you, my sister was and still is a very spirited person. Uh, in fact, in high school, she was a cheerleader, and when she went before I did to UCLA, as you can see right here, she's a Bruin also. She tried out for the cheerleading team and almost made it. She wanted to be the token Asian cheerleader. Uh, but I digress. Uh, my sister is very spirited, and sometimes when she's not winning, she'll say, "Hey, machine, it's time for you to pay off now." And my sister's very smart. She went to UCLA. She was valedictorian of her high school class. She knows that the probability of events are independent of each other. They're not ready to pay off. They just do that occasionally, just enough to keep you there. So my sister is an example of this law of probability. Have you seen that Friends episode when Phoebe went into the casino and she was putting her quarters in, and then she would get tired of playing a particular machine? Excuse me. And she would uh, "Hey, um, no," she would go to and stop playing, and then a grandma came right after her, put a quarter in, cha-ching. So Phoebe called that grandma the lurker because she kept falling around in that episode. That also an example of the understanding that events are independent of each other as far as their probabilities. Now, the rule of multiplication is another rule of probability. It states that the probability of two independent events uh, is calculated by taking those separate probabilities and multiplying them together. So my wife is right here, and these are her sisters. So here's my wife, and here are her sisters. My wife is the middle child. And the question I ask you is this: what's the chance of having five girls? Is it really high or really low? When you're driving around town, and you look in I don't know, look in a Yukon, an SUV, you look at, there's five kids in there. Is it unusual to have five boys, or like my wife has in her family, five girls? Or is that more the norm? If you're going to have five kids, is it usually be five girls or five boys? Or is it more likely to be two boys and three girls, or three boys and two girls, or four and one, or one and four? Well, I think in my practical experience, it's quite rare to have five of the same gender. So here's what it is: it's 1/2 to the 5th power. And so that's a 1 out of 32 chance. It's pretty rare. So this symbol right here means it's 1/2 times 1/2 times 1/2 times 1/2 times 1/2. Because each time there's a 50% chance of you being a male or female. So how do we figure that stuff out? We do it with what we call a Punnett square. A Punnett square is a diagram used to predict genetic cross results. We take the genotype of the sperm, and that's just going to be one letter, and put it over here. And this is the genotype of the eggs. And so we have, remember, they split up because of that first rule of genetics. So in a heterozygous individual, we had a big S and a little s. And so the sperm and the eggs of the male and the female are both heterozygous. And the genotype here is big S and little s. But then these two come together during fertilization. So this one is big S, big S. Over here, this combination is big S, little s, and this one is as well. You got a 1 out of 4 chance of getting a little less and a little less. That's a homozygous recessive. Here's some numbers that will be essential for you to know on the test. The phenotypic ratio is 3:1, and the genotypic ratio is 1:1 for a monohybrid cross. Notice that these pairs of numbers, these sets of numbers, add up to 4 because there are 4 boxes. If you take a look at the 4 boxes of a monohybrid cross, that means one characteristic, both parents are hybrids. For a monohybrid cross, these are the results. And so the reason we call the 3:1 phenotypic ratio is because there's 3 that are smooth and 1 that's wrinkled. And then the genotypic ratio is 1:1. Why? Because 1 is big S, 2 are big S, and 1 is little s. That's what we mean by 1:1 for the genotypic ratio. Also, for a test cross is used to determine unknown characteristics. Let's say we want to know what that second letter is in the dog here. In this dog, we don't know if that's a big B or a little b because it's already black. If it has one big B, it's black. If it has 2 big B's, it's black. Now, the thing is, if this is a big B, then all the puppies of this pair are going to be black. But if this is a little b, then you have a 50% chance of getting a chocolate lab. So this is a chocolate lab. This is a black lab. And here, this is what our prediction is for the puppies. Now, if you have a litter of 10 and you have only one that's a chocolate lab, well, guess what? Then that still has to be a little b because there's no way to get a chocolate lab if this is not a little b. If you have a litter of 10 and all of them are black, then you've got statistically a pretty good idea that this is a big B. Okay, so that's a test cross. Now, obviously, you can't do that with people, but you can do that with plants, and you can do that with, they did it back in the day out on a cattle ranch. They would do it with cows, steer, because they wanted to get the cows that had a certain horn. You know, instead of having the really long horns, maybe you'd rather have a short horn that curves downward so that you can pack more animals together. So that's a test cross. All right, here's the second law of genetics. Mendel's second law says the law of independent assortment. It's also called the dihybrid cross. We're going to take a look at 2 characteristics now. That's why we call it di. But for each of these 2 characteristics, it's going to be hybrid. Hybrid, remember, 1 of each letter. So the law of independent assortment is revealed by tracking 2 characteristics at once. Now, here is the correct one. This is not correct, but what's important on the slide is this more formal definition. It says that the second law, the law of independent assortment, states that alleles of a pair segregate independently of each other. Allele pairs of independently of other allele pairs during gamete formation. So these are not relying on each other. They move separate from each other. This is true if these guys are on separate chromosomes, these characteristics, and we saw that the characteristics that Mendel picked were on different chromosomes. So his mathematics worked out well. But we'll see at the end of the talk that if these characteristics are on the same chromosome, then you have a situation where these are called linked traits, and they will be inherited together. The law of independent assortment says that these alleles for different characteristics will separate independently of each other. Now, this is we're going to do one of these when we take a look at our genetics problems, our sample problems, but you should know the ratio that we're going to take a look at. The ratio that you should know is this ratio, 9:3:1. So now you have 3 different ratios to remember. You have 3:1, you have 1:1, and now you have 9:3:1. Now these add up to 16 because when you do a dihybrid cross, there's 16 boxes. And you have now, instead of a 1 out of 4 chance, you've got a 1 out of 16 chance to have a double recessive organism. And oops, it's that one right there. So this is a little y, little y, because it's a 1/4 chance of getting a little r, and a 1/4 chance of getting a little y, 1/4 times 1/4, multiply the denominators, and you get 1 over 4 times 4, 16. So that's how you get 1 out of 16. Here's another example of interdependent assortment for those of you who have Labrador retrievers, you like dogs. Here's another situation. Actual Labradors are an example of a non-Mendelian characteristic as well. But if you take a look at these numbers, you have 9, 3, 3, and 1. All right, as we close up today, I want to take a look at some conditions where things happen that are not predicted by Mendel's laws. So these are the 6. So let's take a look. First of all, there's incomplete dominance. Incomplete dominance, an example of this is in the plant called the snapdragon, and this results in intermediate phenotypes. Instead of having a red or a white right here, the red and the white doesn't quite go away, it's not quite overpowered by the red, so this appears to be pink. So whenever you have a heterozygote, it's pink. This happens in humans as well. For the situation here with the cholesterol, these are LDL receptors that when in a normal cell, it will have these LDL receptors. If a person who has this condition of hypercholesteremia, they might have no LDL receptors, but you might also have a heterozygous, which would have fewer LDL receptors and so have a milder case of the disease. So this is incomplete dominance, where the heterozygote is between the dominant and the recessive phenotypes. Now, before we go on to the next one, we need to realize that you have 2 genes for most things, for many things. You might have 2 different alleles, or you might have 2 copies of the same allele. But in the population, there are more than 2 alleles. For example, there's eye color. And you have, here's a situation. Someone with brown eyes may have gotten a brown allele from one parent and a blue allele from the other parent. Both only show brown eyes, but there's still green in the population. So how do you get green eyes? Well, there are different alleles in the population. So another characteristic where the characteristic of multiple alleles is significant is the ABO blood type. Those are three different phenotypes: A type blood, B type blood, O type blood. Well, 2 of those are what we call codominant. They don't overpower each other, but they both show up. So a person with O type blood has an O and an O. A person with A type blood has an A and an A, or an A and an O. In this designation, the O is a little I. So if your genotype is OO, you show O type blood. If your genotype is big A, big A, big A, or if it's AA or it's AO, you have A type blood. If it's BB or BO, you have B type blood, but this is the one. If you have an A and a B, neither of these are dominant over each other. And so that's how you get AB type blood. Before we leave this, to successfully do the quiz this time, you need to know that a person with O type blood can give to anyone. We call that the universal donor because in this diagram is also showing where clumping takes place. You go down here, there's no clumping. On the other hand, a person with AB type blood can get blood from anyone. So we call that the universal recipient. And that universal recipient can receive blood from any blood type. Here's another example where Mendel's laws don't quite work because a single gene affects many phenotypic characteristics. So this is just one gene and it causes sickle cell, but as you can see, it causes all kinds of different problems in the body. So that's why sickle cell anemia is such an important disease because it is an example of pleiotropy, where just a change in one gene has many different changes in the phenotype. On the other hand, in epistasis, what happens is that you have to have more than one gene that determines the characteristic. So back to Labrador retrievers, we've seen the brown and the chocolate in a previous example, but there's actually yellow labs. I have a yellow lab. And so how do you get a yellow lab? Well, you get a yellow lab if you are recessive for this second gene. If you're the homozygous recessive of the second gene, masks the first gene. So you get a yellow lab when you have a little e, and it doesn't even matter what you are for the first gene, because to get a yellow lab, it's going to cover that up. That's called epistasis. Over here, it's more common to have a black lab. I have one of those also. It's a 9 out of 16 chance that she would be black. And for the chocolate lab, I mean, this one is little b for the first gene, but not recessive for the second one. So this gene is expressed. So that's epistasis. Now to humans have many different characteristics. Many of our characteristics are what we call multigenic or polygenic. Two or more genes pairs contribute to the phenotype. And so there's all of these different characteristics you can see that are multigenic: height, intelligence, skin color. Here's a really tall guy in Laker colors. Chi Soon, tallest man without a genetic disorder for normal height. Woo! There have been people taller than him, but they have been as a result of different genetic abnormalities like Marfan syndrome, for example. Some people say that President Abraham Lincoln perhaps had Marfan syndrome. But Chi Soon, what does he have? Well, he doesn't have a genetic disorder, but he turns out that he is 7 feet, 9 inches tall. Yes, we could use him on the Lakers. Now, and another example of multigenic trait. This is for skin color. You can see in this situation, you have 3 different genes that attribute to skin color. Now, because you have 3 different genes, you get more, instead of a bar graph, you get more of a curve, more of a continuum. So my mom was pretty light. My dad was pretty dark, and I'm probably a little bit lighter than my dad. I'm a little bit on the darker side over here. And so I'm a combination of my mom and my dad. But as far as height goes, I'm an inch taller than my dad was. I'm 5'7". My dad was 5'6". My mom was 4'10". So I really should have been much shorter, you would think, statistically, but I turned out taller than all of them. So I was over here. So many of our characteristics are multigenic. Many of our characteristics are polygenic. Many of our characteristics are dependent on more than one gene. And then the last one I want to take a look at today is phenotypic plasticity. With phenotypic plasticity, it says many traits are affected in varying degrees by both genetics and environmental factors. I said I'm a little dark, but actually during the summer, I tend to be a little light. In fact, my feet are very light. In fact, so light that my sisters would make fun of my feet because they are so light compared to my arms. What's up with that? Well, my arms are exposed to the environmental factor called the sun. And so the melanin that my dad gave me produces a protective situation. And so I'm a little darker there, but I don't get sunburned. So that's a good thing. That's phenotypic plasticity. People laugh when I say I'm like a chameleon. I have many different colors. And I can be. In December, I'm a lot lighter than I am when I'm working on the yard in July. So phenotypic plasticity. All right, so those are some ideas about genetics that I'd like you to know. If we take a look at, uh, well, now it's time for us to practice some genetics problems, but I'm going to do that in a different talk. So that you have a chance to take good notes, but there'll be a different presentation where we go through some practice genetics problems together and see how to solve those. Okay. All right. I'm Mr. Vallejo. Thanks for coming to biology class, and we will see you next time. All right. Bye-bye.